Hello everyone, my name is Lindsay Hopson. I am a second year bioinformatics master's student, and I'll be giving a tutorial on how to navigate through Matlab to perform machine learning analysis on metagenomics data. So first I'm going to go over the data that we used for this analysis. Our group analyzed fecal samples collected from wild-type mice and TGF beta signaling deficient mice. These mice have a deficiency in a signaling pathway that ultimately leads to colorectal cancer development. These mice were either treated with fluorouracil, which is a chemotherapeutic drug, or phosphate buffered saline, which is a control solution that does nothing. And fecal samples were collected from these mice at 3 different time points: before treatment, during treatment, and then after treatment. And so our group did bioinformatic analysis on these fecal samples and ultimately created this abundance profile table that you see. And so in this table, it shows the different samples that were collected in columns. It shows the different samples, and then it also shows the organism name here. It also shows extra information such as lineage and GenBank reference number. And so we then calculated the relative abundance of the different species within these different samples, and that's what's shown here, the relative abundances. And so we use this data to then perform machine learning analysis to answer these 3 questions. Is there any signal differentiating between a TGF beta deficient mouse and a wild-type mouse before treatment? So we want to know, is there any difference in microbiome composition before they're treated with anything? We want to see if there's any differences in these mice types. And then if there is a difference, which brings us to question 2, if there is a signal that differentiates between these 2 mouse types, what are the important predictors? So what are the important bacteria species that contribute to this signal that differentiates between these 2 mouse types? And then lastly, last question is there any signal differentiating between wild type before treatment and wild type after treatment? So are we able to see a signal from the gut microbiome between the same type of mouse, but one is given treatment and one is not given treatment? Can we see a difference in their microbiome? So we're going to be using machine learning to see if we can predict these 3 different questions. So in the step-by-step tutorial, I go through how to download Matlab and how to download the statistics and machine learning toolbox. For the sake of the video, I will not go through that, but it's shown in the tutorial. Also, I go through how to manipulate this table that we'll be using. So for Matlab, we want to remove specific data that's not necessary to be able to answer the question. So in the written tutorial, it shows on how I manipulate this table, but I'm not going to go through, I'm not going to exactly show you. I'll just quickly talk through what I did and then show you the final table that we'll be importing into Matlab. So because we want to answer a specific question, is there any signal differentiating between TGF beta deficient mice and wild type mice before treatment? We only want before treatment mice. So we're going to actually remove all data that was taken during treatment and after treatment. So the DTs stand for during treatment, samples collected during treatment, and then after treatment is AT. So we're going to delete those samples, the wild type before and after, during and after treatment, and then we'll delete the TGF beta during and after treatment samples. And then we're next going to delete lineage and GenBank reference. We don't need this material to do the analysis, and we're going to keep these organism names. And then lastly, we're going to actually transpose all the data. Matlab specifically likes the format so that samples are in rows and the variables are in the columns. And so when we finished manipulating the data table, it will ultimately look like this. So again, the data was transposed. The samples are now in rows, and the organisms are now as columns. And so this is what we'll be uploading into Matlab. And so we saved this file as WTV TGF. And so now we can import this into Matlab. So we're going to go ahead and open Matlab. So I've opened Matlab, and this is the page, like the main page. And so we want to import our data. Also, you want to make sure that you have the statistics and machine learning app installed. Yeah, the toolbox was already downloaded on my computer, so it doesn't have any downloading options. But for people who don't have it downloaded, that's how you would download it. Like, look it up in the search and then follow the installation rules. It's just clicking installation and then it'll download. So that's that. So now we're back to the main page, and we want to upload our table into Matlab. So first you want to access the folder that has the table. Since I used Matlab before, this is already like automatically filled in and shows the folder. But for new users, it'll be blank. So you want to click on this little folder here and select download. Well, this is where I have my table. So it's in downloads, and then I made another folder called Matlab demo. Click on that, select open, and then it'll show like the path of the folder, and then all the stuff that's in my Matlab demo folder is in current folder panel over here. So the table that we created was wildtype _ v _ TGF. So we're going to select it and drag it into the command window, and you'll see like this little code pop up. And then next, the import wizard will pop up, and you want to make sure that output type, the table is selected. And then you want to make sure that all the data is highlighted. And so then you want to push import selection, and you'll see that the table is now in the workspace. So you can just exit out of this. The table is now in the workspace, so now it's imported into Matlab, and we can start running our analysis. So again, our first question is there any signal differentiating between a wild type and a TGF beta deficient mouse before treatment? So we want to classify the type of mouse and create a model that classifies mouse type correctly, or the most accurate. So we're going to go to apps, go to classification learner, or if you don't see it immediately right here, you can also just click this drop down arrow, and it'll have all your apps, and you can click classification learner that way. So this is the classification learner window. You want to select new session from workspace, and then under data set variable, you want to make sure that the table that we're going to be using is selected. If you've already imported different data tables, then you'll see the other ones here, but since we only have one, this is only one available. Then under response, you want to select from data set variable, and then you want to make sure that our response variable is selected, which is mouse type. And so our predictors would be all the different bacteria species here. That's what the model will use to predict the response variable, which is mouse type. Over here, we're going to leave it as default. Cross-validation helps determine the accuracy of the model. So we're going to leave it as default. Start session, and then make this bigger. Then we want to train our data on all the models to see which model best predicts the response variable. So we are going to select all for training all models, and then we're going to select train. And so on the side here, you can see all the different type of models loading, and then it shows the accuracy on how well the model is able to predict the response variable. And so in the written tutorial, I've provided different resources to help people learn like what k-nearest numbers, the KNN means, these different type of models. Like, there's a lot of information about what these different models do, and I would suggest like reading about them to understand what they do. So here, everything is finished running. So here it shows that the simple decision tree was the most accurate model. It predicted the response variable, and it predicted everything correctly. There's 100% accuracy. You can look at other models and their accuracy. And so if you want to look at how well the model predicted mouse type, you can look at confusion matrix, and you can see the confusion matrix and how it was predicted. And then you can also look at ROC curve, and you can export both of these type of models by over here, export model, and stuff like that. So that answers our first question. There is actually signal between wild type mouse and a TGF beta deficient mouse before starting any treatment. So now we want to answer our second question. Is there what are the important predictors? What's causing, or what organisms contribute to this signal that differentiates between a wild type mouse and a TGF beta deficient mouse? So we are going to leave this classification learner and go back to like the main Matlab page. And then we are going to use this code called ensemble bagged M. So if you double click on it, it'll open, and it'll there's like notes in here, and it creates a random forest tree, and then calculates the important predictors. And so we're going to run this code. You can either run it by just double clicking the code, and then it'll pop up. You can select run, and it'll run, and it'll produce the important predictors plot. And here it shows that E. coli strain NCTC 13441, that is the most important predictor for differentiating between wild type and TGF beta deficient mice. And if you want to see other important predictors, like here, in the code, we only specify the top 5. So if you want to specify top 10, you can list it there. Or if you want to change the title of the plot, you can change it by specifying it in the code. Important. And then rerun the code. So now it shows the top 10. Also, you can also just run the code in the command window. Just by typing in ensemble _bagged, enter, and it computes the important predictors. So I went through questions 1 and 2, and I'm not going to go through question 3 due to time. Question 3 can be solved similar way as question 1. You have to manipulate the original data table, remove data that won't be necessary to answer the question, import the table into Matlab again, use the classification app, and select the most accurate model. If you have any questions, please feel free to reach out to me. Thanks for watching.